welcome back to the class. In our last class on this chapter, which is called as the molecular basis of inheritance, we understood how well the DNA structure is designed. Yeah, it looks like well, but at the same time it looks complicated. It also looks you see that there are a lot of differences between DNA and RNA, though they are all made up of the polymer or polynucleotide chain, right? Now, when we have a double-stranded DNA in front of us, and this double-stranded DNA has all the information in it. Obviously, the first thing that comes to our mind is, where is this information stored? There has to be some kind of cryptic language which this DNA has. And if you just look at this, the phosphates are the same. Whether you are talking about the DNA or the RNA, the phosphates are the same. The sugar is almost the same, right? So there you do not see any difference. So where exactly you see the difference? Now all the genetic information is coded in this nitrogenous basis. The arrangement of these nitrogenous bases is so profound that the three consecutive nitrogenous bases actually codes for one amino acid, and so many amino acids one after the other will give rise to a protein. So basically the whole of protein is given in a form of the A, T, G and C code. Yes, to understand this we took a while, but once we have understood how this really works, there's a lot of advancement in understanding the molecular basis of inheritance. So basically we have DNA, and this DNA is so beautiful. It is double stranded. Everything is the same other than the nitrogenous basis. There are two strands. One of them is coding for a protein and the other one is not coding for a protein, right? So one of them, the exact sequence, please do remember that in our last class we learned that they are complementary to each other, and because they are complementary with each other, obviously one of them codes for a protein and the other one does not, right? Now the right particular fragment of the DNA is getting converted into a protein. But of course this does not happen in a single step. The DNA which is there gets read into another polymer which is our RNA. Very easy to get a copy of that, right? So you replace T's with U's. So suppose there is on one of the DNA strand, let me say I have A, T, G, C and A. It's very easy for me to make another RNA molecule, and this RNA molecule would be, if there is A, there would be uracil, right? So adenine with uracil, thymine with A, G with C, C with G and A with U. So this is the RNA molecule. So what we see is, we see that one strand of DNA is read as a RNA molecule. Now RNA molecules is not very long, so what happens is this RNA molecule which is formed comes out of the nucleus and comes into the cell, and once it comes into the cell, you see that this RNA is recognized by the ribosomes, and the ribosomes reads this RNA, okay, and gets the right amino acid. Suppose this is coding for, let me say, lysine, an amino acid. Lysine comes and gets added here. So you see that the RNA is read and the right protein with the perfect proper amino acid is made. Okay, now this is what happens within our cells. So genetic information coded as DNA, converting it into RNA, converts that into protein. It's not actually convert, I would say it as reading, that would be a better term. So this conversion of reading of DNA to RNA is what we call as the process of transcription, and then we have RNA to protein, which is called as translation, right? So, but please do remember that DNA also has the ability to make some more DNA, which is called as the replication. Okay, so basically for the DNA to efficiently act as a genetic material, it is able to carry out replication, can give rise to RNA by the process of transcription, and then it also has RNA has the ability to give rise to a protein, and the whole of molecular basis of inheritance is dependent on these processes of replication, transcription, translation. And this is a very important process in molecular biology, which is called as the central dogma. This is called as the central dogma, right? So the whole of molecular biology, the whole of life is revolving around these processes. Now, if this is so important, DNA is important, RNA is important, we have proteins which actually carries all the message, so proteins are important. This DNA is present inside the nucleus, which was found quite early, even before Mendel found out the factor. So we know by majority of these things. But how is this DNA present inside a small nucleus? Right? Please see that we have already seen the mathematical aspects here. You see that the difference between two bases is 0.34 into 10 to the power of minus 9 meters, nanometers, right? That is the distance. Now we know that our haploid DNA has 3.3 into 10 raised to the power of 9 base pairs. Basically is the measure which we use. I told you in the last class that I will tell you in this class. Since DNA is a double helix, you have bases which are paired, so we call it as base pairs. Okay, so we have 3.3 into 10 to the power of 9 base pairs in a haploid cell. Now if that is the condition, just imagine that I want to fit the entire DNA molecule into a nucleus which I can't even see without a microscope. Now how is that possible? Right? Now that itself is very astonishing fact. Though we talk about the DNA being very important and all that, you see that the DNA is not just present as thread-like structures as we see here in the structure. Okay, they are kind of organized into a much more complex structures. What do we mean by complex structures? See, we have already seen that DNA are acids, nucleic acids, deoxyribose nucleic acids. They are all acids, and these are because of the presence of the phosphate group. Okay, DNA is negatively charged. Agree with this? Basic chemistry. So we have the phosphate group which is negatively charged, and DNA itself is a polymer, it is an acid and it is negatively charged, and now this is not very, if I leave all of this together it will form a clump. Therefore, you see that in the first place this DNA is bound to a positively charged proteins. This positively charged basic proteins are what we call as histones. Okay, it's called as histones. Please do remember that these are proteins. Now how can a protein become positively charged or negatively charged or neutral charges? That is because, please do remember that this protein is also a polymer, and this protein is a polymer of amino acids. Now these histones are rich in 2 amino acids. One amino acid is called as the lysine, and the second one is what we call as the arginine. Okay, the first one is called as lysine or arginine. We write it as lysine or arginine, ARG or LYS. Now these amino acids, both of them are positively charged, and the entire histone proteins are very rich in lysines and arginines, and therefore you see that they are basically basic proteins, basically positively charged proteins, right? Okay, now histones, though we just tried that they are one group of proteins and we write it like this, you see that its structure is not so simple. They are called as octamer. Octa is 8, mer is one single unit. So one single unit of a histone consists of 8 parts. Okay, and so you see that all the 8 of them, 1 2 3 4, and then you have at the base another 4, all this are kind of joined together. Okay, the negatively charged DNA, you see that it kind of comes from one here, goes one round, comes out here, and it kind of gets wound around the histone. Okay, it goes around the histone. Now it is seen that one single nucleosome, if it starts from here until the time it comes here, it can have around 200 base pairs of DNA helix. So 200 DNA base pairs of helix will go around the nucleosome. Yes, so you see that the size of the DNA, which was very long, becomes quite small. Not very small though, it becomes small. Now, just to see that the DNA does not come out easily, okay, from this, you see that there is one more part of histone which is called as the linker, histone 1, H1, which is called as also as linker histone. Now, this will kind of see to it that it is held together. Something like, you have already wound the DNA, just to see that they are staying together, you put a pin or something like that. Now, this linker histone or the H1 histone will bind the DNA onto the octamer histone. Okay, and this is something which is very important. Okay, very important, because from here this gets wound up to give you a chromosome structure. So people have seen this kind of structure under the microscope, and the structure looks very much like this. It looks as if you have a huge large string, and on the string you have lot of beads that are present here. Now this is actually called as the beads on the string model. All the beads here, they represent the histone core. Then you see the string there in between, that would be the linker DNA, and each one of them would be held together. It would look like a bead on a string model. Now with this, you see that the whole of the DNA, long DNA, becomes smaller. Now this structure where the DNA is wound to the histone molecule, we call that as the nucleosome. What do we call that as? We call that as the nucleosome. Now generally a nucleosome is a complex which has histone and a DNA which is around say 200 base pairs. Okay, so approximately 200 base pair length of the DNA. Okay, now this has the DNA which we call as the chromatin. Okay, so the threads that you see there is what we call as the chromatin, and these are the nucleosome structures. Now, how many of these beads, how many of these strings can you see in a mammalian cell? Now that is huge. That is huge. Now from this beads that you see that they are further coiled, okay, and further condensed. Now here I would like to give you one very simple example. Yeah, it's been quite some time that we don't have landlines at home, but yes, if you have seen a landline, which I'm sure you would have a connecting wire to that phone and the receiver, right? So you see that there is this coiled wire. You keep on winding the coil, you see that they kind of intertwine, and this lengthy wire would become very short. So even in our DNA you have this chromatin. Now this chromatin, which is bound to histone molecules, would wind again, coil up, such that their length becomes very, very small, and it becomes so small and so condensed during the metaphase stage of the cell. And at this point in the metaphase stage of the cell, we call them as the chromosomes. That is, the chromosomes are the DNA genetic material which are the condensed chromatin. Condensed chromatin is condensed nuclei, condensed histones with linker protein, which again condenses to give you the smallest, shortest, coiled structure, which is the chromosome. Okay. Now you might think that does histone only will bring about all this coiling? No, only for the chromatin. Chromatin is that bead structure, bead and string structure. There is requirement of histones, but again, to coil it further to chromosomes, we don't need histone proteins. In fact, those group of proteins are called as the non-histone chromosomal proteins. We call them as non-histone chromosomal proteins. So you see that generally in a nucleus, certain chromatin is very loosely packed, some of them are tightly packed. Okay. Now the tightly packed one, so we have the tightly densely packed chromosomes. They're not the completely separated part from the chromosomes, and if suppose I state them, you see that they appear darker. And such regions is what we call as the heterochromatin. Such regions of the DNA densely packed on the chromosome, we call it as the heterochromatin. However, certain regions which are actively involved in making proteins, making RNA, okay, trying to control something, and all of them, they stain light. Okay, they stain light, and we call them as the euchromatin. Now it is assumed that the euchromatin is the region which actually codes for the proteins required by the cell. Okay, but we say that it is the transcriptionally active chromatin, and heterochromatin is inactive. But however, there are many exceptions to this. Many exceptions are seen, okay, and especially some of the heterochromatin which is not labeled properly, we see that they lead to the production of some other proteins which are not required by the cell and might lead to cancer. Okay, but however, for our understanding, let us just know that euchromatin is the one which is transcriptionally active chromatin, giving rise to RNA and protein, whereas heterochromatin is densely packed, does not undergo transcription or translation. Right. So this is how it was in talking about how the DNA is actually packed. We call that as the nucleosome structure and so on. Okay. So once we know about how the DNA is and what the DNA is function is, okay, we should get to know how exactly the DNA was found out as the genetic material, right? That itself is something which is very, very interesting story. So we will start off thinking about how this DNA is considered as the genetic material. Okay, so Let us start off learning about the search for genetic material. So I told you in the last class that sometime in 1869 there was Miescher who discovered the presence of acids inside the nucleus, right? And then was Mendel's study sometime in the late 18th century, 1850, after 1880s. Mendel proposed his theory of inheritance. He said that there is something which is there inside the cell. He didn't say nucleus, of course, and he said that there are something called as the factors, and these factors will carry the information. So this was told by Mendel, and for a very long time people thought that that yes, Mendel's work is Mendel's work, Miescher work is Miescher work, and it was not very easy for them to connect and tell that yes, DNA is the genetic material. Okay. However, in 1902, in our last class, I was telling you that there were a lot of experiments that were done, and in with lot of series of experiments that was done, we realized that DNA is the genetic material. Okay. In 1926, people really wanted to find out where are these genes, where are the proteins coming from. Okay. At what form and what stage would they go, and many things like that. Okay. But by that time we have already seen by the Sutton and Bowery's discoveries and Morgan's experiments, that we had kind of realized that chromosomes are something which is very important to determine the trait. So, and chromosomes were inside the nucleus. So this was kind of trying to link that are chromosomes carrying the genetic information. Okay, and people really started working on that lines. Okay, now sometime in the year 1928, there was this very famous scientist. He is remembered every day in the field of biology, because of which he kind of changed the whole field of biology. Okay, very simple experiment, but a wonderfully planned experiment. His name was Frederick Griffith. Frederick Griffith. Now this Frederick Griffith was a wonderful scientist, lot of insight, working with organisms, working with mice and many others. Now he was working with one organism, bacterium, which was called as Streptococcus pneumoniae. Okay, now this is a bacterium which is which causes pneumonia. Okay, now he was growing this bacteria, and he saw that the organism actually changed a lot. Okay, he kept growing them in the, we call that as a Petri plate, glass plate on which the bacteria are allowed to grow. So he keeps that organism and keeps it for a very long time, and he saw that many of them were trying to change their physical form, and very soon he saw that many of them were changed completely. Okay, so gradually from one form to other form. Okay, so basically when he grew them on the culture plate with the Petri plate, okay, he saw that some of the colonies had a sheen on them. Okay, and they were called as the S colonies or the shiny colonies. And now there was one more which was not shiny but if you just look at them, you see that they had some small dots present on them, which made their appearance slightly rougher. Okay, and he called this as R. So this S strain bacteria, you know, they are smooth. This is not smooth. Now this becomes smooth because of something. It cannot be just like that. So this gets that shine, it loses its roughness because they have a mucus coat on them, and this mucus coat is nothing but the polysaccharide. Okay, so there is a polysaccharide mucus coat on them, whereas the R strain will not have that. Okay, now if by chance mice are infected with this S strain, you see that they cause infection. Okay, so we call this as the virulent strain. Virulent is basically for virus, which causes infection. Virulent is basically for organisms which have the ability to cause an infection. So this one, if I put S organism into mice, this S will go inside, grow swell, causes pneumonia, and the mice will die. Whereas if it is R strain, you see that they are avirulent. Avirulent meaning that they do not cause pneumonia. Okay, so basically what he was trying to do is, he was trying to take both the strains and inject them into mice, and he wanted to see what would happen. Okay, so he has already found out that if I take a strain and I inject them into the mice, then what would happen? The mice die. Now if I take the R strain and I am going to inject them into the mice, you see that the mice live. Okay, now so this was the first observation. Now what he did was, he tried to kill them. Okay, you can take these organisms and you can kill them, obviously. Okay, heat it at a high temperature. Now if I take killed organisms, so I've killed the organism already, and then I inject them into the mice, what do you think it would happen? Because they are killed, the mice live. Perfect sense, right? Okay, now what he did was, he took R also and he killed them, right? And then he injected them into the mice, and when he did so, even then the mice lived. Pretty understandable, nothing much in this, right? Quite understandable. But scientists won't stop at that. They would like to experiment a lot with whatever combinations they have, and so what they did was, we know that R does not give disease. So what he did was, he took the R, please see that it is not killed, and then he took the S strain that is already killed. So one is already killed, the one which is virulent is killed, and a virulent one is taken together, and now they were given and injected into the mice. Now what should happen? Mice should live, right? Why should mice live? Because this alive one is avirulent, the killed one is virulent, but it's already killed, so it should not cause disease. So this was the whole principle. But then what Griffith observed was completely different. He could see that all the mice got the infection and they were all dead. Okay, big confusion. So if R is giving the disease, it means that this R structure is changed, physical structure is changed. So from avirulent it has become virulent. Yes, so what is happening here? So he got to know that this R, from being a good bacteria, it has become transformed into a bad bacteria. from a avirulent strain, it has transformed into a virulent strain. And then he called that entire principle as the transforming principle. What did he call it as? He called it as transforming principle. And according to this, he said that something in the S-killed organism, something, okay, so something in S-killed bacteria is moved into our bacteria, and this, once it's moved into R bacteria, this has changed the R bacteria into a S bacteria. So there is no R, there is S, and this S, why does it become S? Please do remember, it's because they can synthesize the smooth mucus polysaccharide layer, so they are able to synthesize that coat, and that causes virulence. Okay, so this was the transforming principle which was discovered by Griffith. So what he said was that something goes, of course he did not tell that it was DNA. So people started searching for what is that transforming principle after this. Okay, so in 1928 these studies were done. But this whole of transforming principle is very, very important in biology. You might wonder why it does not tell us anything about the DNA, right? So why is it so important? There are two important things here. The first thing is it tells us how to frame the experiment. Okay, if I have an experiment, how am I supposed to put that into its right place? That is the first thing. Okay, so there was no need to mix R and S killed here, but he tried to do with that. The second most important thing is his observation, and then the third was about framing a hypothesis and trying to prove that. So all the basic research methodology was very well done by this particular experiment, and hence Griffith's experiment is something that you learn in as a model experiment when you want to do research in biology. So all this happened sometime in 1928 and 1930, and the transforming principle was discovered. But obviously right now we are in search of DNA, the genetic material. So for us that is not enough. Okay, so people started working more to understand the transforming principle, the factor, then chromosomes and so on. And then between 1933 and 1944, there were three important scientists who thought how the proteins are being formed. Okay, their names were Oswald Avery, McLeod and McCarty. So there were three people, Avery, McLeod and McCarty. They thought that the chromosomes gave rise to DNA and DNA gave rise to protein. Okay, now what they worked was completely to understand, I'll write it here, to understand the biochemical nature of this transforming principle. Okay, so what they did was they tried to take out, when they kill the bacteria, we can classify and separate them into DNA, into RNA and into proteins. So instead of taking the S killed, they purified DNA, RNA and protein from the S killed, separated them basically. And then, so there were three tubes here, right? So in one of them they took DNA, In one of them they took the RNA, and then in the other one they took the proteins, and then they mixed our organisms here, and this mixture was injected into the mice, and they saw that this mice lived, this also lived, whereas this died because of pneumonia. Okay, this died because of pneumonia. Why? This is because the DNA which was present in this separated filtrate here was already transforming the R organism, and that caused the mice to get the disease and die. Whereas in the case of RNA, the RNA was digested by RNases, which is present in the organism. Then the proteins were digested by many of the proteases. So thus they discovered that DNA was something that was very, very important for as a transforming principle. So basically it was not completely the tests that was done. They went to understand the biochemical nature of the transforming principle, and later they proved that the genetic material is DNA. So this kind of was the work of Avery, MacLeod and McCarthy. But even then, people were not highly convinced because maybe because the R itself had few enzymes, or their whole of things, was not very clear, and many such things, they wanted few more experiments to be satisfied that DNA is the genetic material. And it was at this time that sometime in the year 1952, a great discovery was done by another two experiments by name Hershey and Chase. Now, Hershey-Chase experiment, in fact, was so strong that people immediately believed that it is because of their work that DNA was kind of proved as the genetic material. Okay, now these scientists were working with bacteriophages. Okay, we know what bacteriophages are. Bacteriophages are basically viruses which eats bacteria. Bacteriophage. Phage is eating, so there was this bacteria eating small viruses, and I'm sure we have already learned how this bacteriophage enters into a bacterial cell, right? And once they go inside, what would happen? So when this experiment was being done, before I tell you that experiment, it's better I would tell you how the viruses work. Okay, so we have let me say a bacterial cell. Okay, there would be a bacterial cell, and on this there would be the virus. The virus which is called as the bacteriophage. Now this bacteriophage has the nucleic material here. Now what would happen is that the nucleic material is sent inside the bacterial cell. Okay, that is sent inside the cell, and once inside the cell we do see that there is a growing of this DNA. This DNA would start replicating. So that is there. Anyway, forget about that. So there is this DNA which would start increasing in number, right? And later in the next phase you see that there is a bacterial cell, and there would be small viruses which would be formed here, which is called as the packaging phase. The packaging phase, and from here you see that when the bacteria is weak and there are enough of viruses inside the body, you see that they break open the cell and the viruses are released. This is how it would happen. So basically this is how the bacteriophages infect bacteria. But now let us see what Hershey and Chase did. It's very pretty much the same, but the only thing is that they were growing viruses on a medium, and what they did was they grew it in a medium. They grew it in a medium that contains radioactive phosphorus, and radioactive sulfur. Okay. So basically you see that there is radioactive phosphorus and radioactive sulfur. So in one of the medium there was radioactive phosphorus, in the second one there was radioactive sulfur. Now why I am writing it as in different lines is just to say that there is no phosphorus in proteins. So wherever there is radioactive DNA, you have radioactive phosphorus. Okay, but no sulfur, because we know the structure of DNA does not have sulfur, right? Whereas few amino acids has sulfur. So if we are growing the bacteriophages in the sulfur, you see that they have radioactive proteins, but they do not have radioactive DNA, right? So I hope you got it. So they were trying to grow two different types of bacteriophages, one with radioactive DNA and the other one with radioactive proteins. Now what they did was they took these bacteriophages and they were made to infect or attach to E. coli. Okay, it gets attached. Now what do we see? Whenever there is infection, the genetic material is sent out, right? Now please do remember that viruses have DNA also, proteins also, DNA or RNA as a genetic material, and proteins. So they don't have cytoplasm, they don't have mitochondria, nothing. So you see that when they inject, they are either injecting the DNA or the proteins, and we would like to see what would happen there later. So you see that the viral coats, as soon as the infection proceeded, the viral coats were removed, and they were agitating them in a blender. Now these virus particles were separated from the bacteria, and we wanted to see how it would look like. Okay. Now if bacteria which was having this bacterial DNA here, radioactive DNA, it is passed on from the virus to the bacteria. Okay. So bacteria which was infected with viruses had only radioactive DNA but not the proteins. Now this shows that only DNA was entering inside the cell and not the proteins. You are getting the point? And thus they got to know that what is moving from the viruses into the cell is not the protein, but it is only the DNA, because there was only radioactive phosphorus and no radioactive sulfur. So this kind of proved to them that proteins did not enter the bacteria from the virus, but there was only the DNA that was entering. And why does DNA enter into the cells and start behaving as if it is living? It is because this DNA was the genetic material, right? So DNA was kind of confirmed to be the genetic material by the Hershey-Chase experiment. Okay, but yes, their way of experimenting was slightly different because they kind of mixed all of them, they centrifuge, they try to find out if there is any radioactivity and all that, and they checked whether the whole cells showed radioactive phosphorus or sulfur. And since all of them showed radioactive phosphorus only, it was proved that the cells, inside the cells, there was phosphorus present. In fact, they also took this, once they infected, they did a centrifuge, right? Now, the centrifuge, when they do, the heavier particles will go down and the lighter will float, right? Now, the one which was supernatant, they took that and they checked whether it has any phosphorus present. No, it did not have any phosphorus, but there was sulfur detected. So, it was kind of very well balanced that this virus has radioactive phosphorus also in DNA, radioactive sulfur also in its coat as a protein, but what was passed on inside the cells was only radioactive phosphorus, and what was not passed out, which remained as a supernatant when centrifuged, was having radioactive sulfur. So, this is how we proved what is our genetic material. Okay. Yes, again, because we are talking about the nucleic acids and DNA and RNA, obviously though both of them are nucleic acids, both of them have the ability to become the genetic material. However, we do not see that both of them are genetic material. DNA is, RNA is not, because of some of the facts, some of the interesting structural differences that has between them, right? So, in our next class, we will try to find out what these differences are and why DNA is the genetic material, and discuss more about the differences between the DNA and the RNA. And if time permits, we will also go to understand a brief description on how DNA replicates. Okay. So, with this, let us just end our class today. So, I hope you have understood. Any doubts, make a note. We will discuss in the live class. See you.